Hello everyone, welcome to my YouTube channel. I hope you all are fine. We are studying BRS Physiology chapter 1 and today's topic is the skeletal muscle. So we will talk about the structure of skeletal muscle and the steps in the excitation-contraction coupling in the skeletal muscle. So let's get started. Okay, so this is the diagram that shows the structure of skeletal muscle. The skeletal muscle, it is made up of thousands of muscle fibers that are surrounded by the connective tissue sheath or fascia. And each muscle fiber is itself, it is made up of myofibrils. And this is the diagrammatic representation of one myofibril, that it contains the contractile proteins, the actin and myosin, which are arranged in basic units known as sarcomere. And this sarcomere is the fundamental contractile unit of the muscle fiber. Okay, so each muscle fiber, it is multinucleate, that is, it has many nuclei and behaves as a single unit. It contains bundles of myofibrils surrounded by the SR smooth endoplasmic reticulum and invaginated by the transverse tubules or D tubules. So we already talked about it in the diagram that each muscle fiber, it is containing thousands of myofibrils, and now each myofibril contains the interdigitating thick and thin filaments. The thick filaments, that is myosin, and thin filaments, the actin, arranged longitudinally in sarcomeres. And the repeating units of sarcomeres, they account for the unique bending pattern in the skeletal muscle and resulting in the striations. That's why the skeletal muscle, it is also known as striated muscle. And the sarcomere, it runs from Z line to Z line. Okay, so in this diagram, it is clearly seen that Z line defines the boundary of sarcomere, because the sarcomere, it runs from one Z line to the other Z line. And we have some other important things to look at. So the thick filament, it contains myosin, and it is present in the A band in the center of the sarcomere. Okay, so the A band is the portion of the myofibril that contains the thick filament. This A band is also called as the dark band. Here we have the thin filament and I band. So the I band is the portion of the myofibril that contains the thin filament, and this I band is also called as the light band. And because of these light and dark bands, the skeletal muscle, it appears striated. And we also have this M line, which is the middle portion of the myosin filament. So M for middle. And edge band or edge zone, it is the portion that contains only the thick filament. So what we learned from this diagram is that the thick filament, it is present in the A band in the center of the sarcomere, and it contains myosin. And myosin, it is a contractile protein, so it has 6 polypeptide chains, including 1 pair of heavy chains and 2 pairs of light chains. And its structure is such that each myosin molecule has 2 heads that is attached to a single tail. The myosin heads, they bind with ATP and actin, and they are involved in the cross-bridge formation. Okay, so cross-bridge formation is something that we will talk about in the following slides. Let's talk about the thin filaments. So in the diagram, we see that the thin filaments are anchored at the Z lines, and they are present in the portion that is known as the I band. And these thin filaments interdigitate with the thick filaments in a portion of the A band. So yes, here, yes, this is the portion of the A band where the thin and thick filaments, they interdigitate. Okay. So the thin filaments contain the 3 types of the proteins, actin, tropomyosin, and troponin. Troponin is the regulatory protein that permits the cross-bridge formation when it binds calcium. So actually troponin, it is a complex of three globular proteins. We have troponin T, troponin I, and troponin C. So the troponin T, the T stands for tropomyosin because it attaches the troponin complex to tropomyosin. And troponin I, the I is for inhibition because it inhibits the interaction of actin and myosin. The last troponin C, the C is for calcium because it is the calcium binding protein. And when it binds calcium, it permits the interaction of actin and myosin. So when troponin C permits the interaction of actin and myosin, we have this cross bridge formation, which is for the contraction of skeletal muscle. And the opposite is the troponin I that will inhibit this interaction of actin and myosin when the muscle relaxes. Okay, then we have the T tubules and the smooth endoplasmic reticulum. So we know that the myofibrils are surrounded by a smooth endoplasmic reticulum and invaginated by the transverse tubules or T tubules. Okay, so the T tubules are an extensive tubular network that is open to the extracellular space because it carries the depolarization from the sarcolimal membrane to the cell interior. See in this diagram that the transverse tubules or T tubules, they are open to the extracellular space and they carry the depolarization to the cell interior. And they are located at the junctions of A bands and I bands. And the special thing about the T tubules is that they have a voltage sensitive protein called the dihydropyridine receptor. And in response to depolarization, the T tubules, they will trigger the release of calcium, which is necessary for the skeletal muscle contraction. Okay, so now where is the calcium coming from? So the calcium is basically stored and it is released from this tubular structure, the sarcoplasmic reticulum. So the sarcoplasmic reticulum, it is the tubular structure that is the site of calcium storage and release for the excitation-contraction coupling. And it has the terminal cisternae that makes an intimate contact with the transverse tubules. And the special thing about the sarcoplasmic reticulum is that the membrane contains the calcium ATPase pump, the calcium pump, which transports the calcium against the electrochemical gradient, that is the active transport of calcium from the intracellular fluid into the sarcoplasmic reticulum. And this keeps the intracellular calcium low. And it also has the calcium that is loosely bound to this protein, that is calsequestrin, and it has this calcium release channel that is called as the ryanodine receptor. Okay, so now we know the basic structure of skeletal muscle. Let's talk about the excitation-contraction coupling. So there are different steps in the excitation-contraction coupling in the skeletal muscle. So the first step is the action potential when it reaches the muscle membrane. This will cause the depolarization of the T tubules. So the depolarization of the T tubules, it causes a conformational change in the dihydropyridine receptor, which triggers the release of calcium from the sarcoplasmic reticulum by opening the calcium release channels, that is the ryanodine receptors in the sarcoplasmic reticulum. And this will cause the release of calcium from the SR into the intracellular fluid. So what happens that the intracellular calcium now increases. Okay, so the next step is the binding of calcium to troponin C. So when the intracellular calcium it increases, the calcium binds to troponin C. So in the resting state, the tropomyosin, it prevents the interaction between the thick and thin filaments. But when the calcium binds to troponin C on the thin filaments, it causes a conformational change in the troponin that moves the tropomyosin out of the way, and the crossbred cycle it begins. Okay, so how the crossbred cycle it works, let's look into this diagram. At first, in this diagram number A, see that the head of the myosin. So we know that myosin molecule, it has a head and it has two heads and a tail. So the myosin head, it is tightly attached to the actin filament, and no ATP is bound to myosin in this state. And here in the second diagram, now the ATP, it binds to myosin head, and this will produce a conformational change in the myosin that causes the myosin head to be displaced or released from the actin. And here in this diagram, the myosin, it is moving towards the plus end of the actin. And now there is hydrolysis of ATP into this adenosine diphosphate, and this high energy phosphate compound, it is released. Okay, so the ADP molecule, it is still attached to the myosin head, and because of the hydrolysis of ATP, the energy that is released, it will allow the myosin to attach to this new site on the actin molecule. So this constitute the power stroke, the force generating stroke. The ADP is released and the myosin is returning to its rigor state. So this cycle, it repeats as long as the calcium is bound to the troponin C. Each crossbridge cycle walks myosin further along the actin filament. So whenever the myosin heads, they bind to actin, they use the chemical energy from the breakdown of ATP to generate a pulling force against the actin filaments, then detach and prepare to bind again. So this repetitive process of binding, applying force, and unbinding, it is known as the cross bridge cycle. Okay, so how the muscle relaxes? Relaxation occurs when calcium is reaccumulated by the calcium ATPase pump. That is the sarcoplasmic calcium ATPase pump, and intracellular calcium concentration. It will decrease. Calcium is released from troponin C, and tropomyosin again blocks the myosin binding site on the actin. As long as the intracellular calcium concentration is low, the crossbred cycling cannot occur. Okay, so that is a summary of what we discussed. So excitation, contraction, coupling. In simple words, the action potential, when it reaches the muscle membrane, there is depolarization of the T tubules, which will open the calcium release channels in the sarcoplasmic reticulum. And there is increased intracellular calcium. This calcium binds to troponin C, the crossbred cycling, it occurs, and again, the calcium is reaccumulated by the smooth endoplasmic reticulum, and the muscle relaxes. Okay, so an important point to remember is that following calcium release, the sarcoplasmic reticulum pump, the calcium ATPase pump, it has been pumping calcium back into the sarcoplasmic reticulum. So if an other action potential it arrives soon after the first, calcium will increase. And if no action potential arrives, calcium will decrease and cross bridging will stop. So on the slide, I just skipped two things. One is this line that in the absence of ATP, there is a state known as the rigor, and it occurs after death. So when there is no ATP, the myosin and actin, they adhere to one another, and this results in the stiffening of the muscles, because when the ATP it is present, it will attach to myosin head and myosin will detach from actin. But when there is absence of ATP after that, these two filaments, actin and myosin, they remain tightly attached to one another, and this will result in the muscles to stiffen. And the second thing is tetanus. So it is one of the bacterial infection caused by a bacterium known as Clostridium tetani. What is its mechanism? So in this condition, what happens that the muscles they remain contracted, especially the muscles of the jaw and neck. So the mechanism is, we know that a single action potential it causes the release of a standard amount of calcium from the sarcoplasmic reticulum and produces a single twitch. However, if the muscle is stimulated repeatedly, as in tetanus, more calcium is released from the SR, and there is a cumulative increase in the intracellular calcium. And because of this, the cross bridge cycling, it continues and the muscle does not relax. So we call this condition as tetanus. Okay guys, that's it for today. Thank you so much for watching. Like, share and subscribe to my channel. I'll see you in the next video.